Good day, students. In today's lecture, we will be discussing the rest of the steps of meiosis 1. In the previous lecture, we have discussed till the prophase 1 of meiosis 1. You must be remembering that meiosis is divided into two steps, meiosis 1 and meiosis 2. Right now, we are talking about meiosis 1, and in meiosis 1, we have divided into four steps again, that is prophase 1. We have also discussed prophase 1, and just to remind you, prophase 1 is the longest phase of meiosis process, and it is further divided into five steps, that is leptotene, zygotene, pecitine, dikinesis, diploidine. And the order is like first step is leptotene, second is zygotene, third is pecitine, next step is diploidine, and the last step of prophase 1 is dikinesis. And each phase of prophase 1 has a characteristic feature or characteristic property, like leptotene is characterized by formation of the conversion of the chromatin fiber into chromosomes, and since a diploid cell contains two chromosomes of same kind and they are called as homologous chromosomes, they approach each other, and in zygotene the two homologous chromosomes or the homologous chromosomes of a pair join together to form synaptonymal complex. In pecitine step, these homologous chromosomes undergo crossing over, in which segments of chromatids are exchanged between the non-sister chromatids. In the diploidine stage, what happens that the synaptonymal complex starts dissolving, that is, the homologous chromosomes start separating from each other, but at some places the non-sister chromatids remain associated or attached, and that point of attachment is called as chiasmata. While in dikinesis, the chiasmata shifts towards the end and the nucleolus and the nuclear membrane degenerates. Now, we will be talking in today's lecture about the remaining steps, that is metaphase 1, anaphase 1, and last is telophase 1. So let's start with metaphase 1. We have to discuss these 3 points today. So first of all, let us talk about the metaphase 1. As we have studied during mitosis, what happens is that during metaphase, that the chromosome get arranged at the center of the cell, which is also known as the equator of the cell. So here also the homologous chromosomes of a pair get arranged at the equator of cell, and since in a homologous chromosome, in a homologous pair, two chromosomes are present, therefore it leads to formation of double metaphase plate or a double wall. Also, spindles are formed. Obviously, the spindles will arrange the chromosomes at the equator. So, spindle fibers are formed. They start forming at the end of the prophase 1 itself. So, here they are completely formed and get attached to the chromosomes. Remember, bivalent consists of two chromosomes or a pair of homologous chromosomes. And this arrangement of bivalence at equator of cell leads to formation of the double metaphase plate or the double wall, because two chromosomes are present in a homologous pair. If we draw it diagrammatically, it will look like this. So for example, this is the cell and these are the spindle fibers. So we can see this is the cell membrane and this is the metaphase plate, which can be called as double metaphase plate. And these are can be also called as the bivalence or the homologous pair of chromosomes. Now, after metaphase 1, the next step is anaphase 1. The third step is anaphase 1. Like in mitosis, during meiosis also, the chromosomes separate. The difference is that in case of mitosis, the sister chromatids get separated and behave as daughter chromosomes. In anaphase 1 of meiosis 1, the homologous pair of chromosomes separate. And this is the reason behind formation of haploid cells as a result of meiosis 1. So what happens during anaphase 1 is that firstly, the chiasmata, which is the point of association between the non-sister chromatids of the homologous pair, that chiasmata disappears, which means that the homologous chromosomes are now completely separated from each other, and now they move toward their respective poles or opposite poles and form two group of haploid chromosomes. So firstly, we should say that chiasmata disappears, which signifies that the homologous chromosomes are now completely separated from each other. And now each is known as called as the dyad. Remember, dyad signifies that in each chromosome two chromatids are present. If we talk about a bivalent, since it is having two chromosomes and each chromosome is having two chromatids, so in totality a bivalent has four chromatids, therefore it is called as tetrad. Now since the homologous chromosomes have got separated by disjunction, disjunction means they have got separated. Firstly they were joined, now they have disjointed, they have separated, and now each chromosome is called as dyad. That means each one is having two chromatids. Now the chromosomes, separated chromosomes of homologous pair move toward opposite pole. And this happens due to shortening of the spindle fibers which are attached to these chromosomes at centromeres, and thus two haploid groups of chromosomes are formed at each pole. Now we can see it diagrammatically here. Now the fourth step of meiosis is telophase 1. During telophase 1, the chromosomes elongate now again to form a chromatin-like network. The nucleoplasm, nucleolus and nuclear envelope reappears. And the result of telophase is that two haploid daughter nuclei are formed. And each nuclei is accompanied by its own set of cytoplasm and cytoplasmic organelles. So, after end of telophase 1, two haploid daughter cells are formed. This is the major difference between mitosis and meiosis is that during mitosis, the chromosome numbers remain same, that if the cell is diploid, the daughter cells formed after mitosis again are diploid. But in case of meiosis, the parents are diploid, but after the end of meiosis 1, the daughter cells formed are haploid. So it will lead to formation of two haploid daughter cells. These are the two haploid daughter cells. Now, after meiosis 1, the two haploid daughter cells are formed. Now, the second part of meiosis, that is meiosis, will start, and in between the two meiosis, interphase may be or may not be present. And even if the interphase is present between meiosis 1 and meiosis 2, then it will comprise only of G1 and G2 phase. There will not be any synthesis of DNA or replication of DNA. So let us discuss meiosis 2. We will be talking about meiosis 2 in the next lecture. This is all for today. Have a good day.